Monoamine oxidase A, also known as MAO-A, is an enzyme that in humans is encoded by the MAO-A gene. This gene is one of two neighboring gene family members that encode mitochondrial enzymes which catalyze the oxidative deamination of amines, such as dopamine, norepinephrine, and serotonin. A mutation of this gene results in Brunner syndrome. This gene has also been associated with a variety of other psychiatric disorders, including antisocial behavior. Alternatively spliced transcript variants encoding multiple isoforms have been observed. Structure. Gene. Monoamine oxidase A, also known as MAO-A, is an enzyme that in humans is encoded by the MAO-A gene. The promoter of MAO-A contains conserved binding sites for sp1, GATA2, and TBP. This gene is adjacent to a related gene, MAO-B, on the opposite strand of the X chromosome. In humans, there is a 30-base repeat sequence repeated several different numbers of times in the promoter region of MAO-A. There are 2R2 repeats, 3R, 3.5R, 4R, and 5R variants of the repeat sequence, with the 3R and 4R variants most common in Caucasians. The 3.5R and 4R variants have been found to be more highly active than 3R or 5R. In a study which did not examine the 2R variant, studies have found differences in the frequency distribution of variants of the MAO-A gene between ethnic groups. Of the participants, 59% of black men, 54% of Chinese men, 56% of Maori men, and 34% of Caucasian men carried the 3R allele, while 5.5% of black men, 0.1% of Caucasian men, and 0.00067% of Asian men carried the 2R allele. The epigenetic modification of MAO-A gene expression through methylation likely plays an important role in women. A study from 2010 found epigenetic methylation of MAO-A in men to be very low and with little variability compared to women, while having higher heritability in men than women. Protein. The gene encodes a monomeric protein which shares a 70% amino acid sequence identity, as well as conserved chain folds and flavin adenine dinucleotide (FAD) binding site structures, with MAO-B. However, MAO-A has a monopartite cavity of approximately 550 angstroms, compared to the 290 angstrom bipartite cavity in MAO-B. Nonetheless, both proteins adopt dimeric forms when membrane-bound. The C-terminal domain of MAO-A forms helical tails which are responsible for attaching the protein to the outer mitochondrial membrane (OMM). MAO-A contains loop structures at the entrance of its active site. Function. MAO-A is a key regulator for normal brain function. It is a flavoenzyme which degrades amine neurotransmitters, such as dopamine, norepinephrine, and serotonin, via oxidative deamination. It is highly expressed in neural and cardiac cells and localizes to the outer mitochondrial membrane. Its expression is regulated by the transcription factors sp1, GATA2, and TBP via the CAMP pathway in response to stress such as ischemia and inflammation. Clinical significance. Cancer. MAO-A produces an amine oxidase, which is a class of enzyme known to affect carcinogenesis. Chlorgeline, an MAO-A enzyme inhibitor, prevents apoptosis in melanoma cells, in vitro. Cholangiocarcinoma suppresses MAO-A expression, and those patients with higher MAO-A expression had less adjacent organ invasion and better prognosis and survival. Cardiovascular disease. MAO-A activity is linked to apoptosis and cardiac damage during cardiac injury following ischemic reperfusion. Behavioral and neurological disorders. There is a link between low-activities forms of the MAO-A gene and autism. Mutations in the MAO-A gene results in monoamine oxidase deficiency, or Brunner syndrome. Other disorders associated with MAO-A include Alzheimer's disease, aggression, panic disorder, bipolar affective disorder, major depressive disorder, and attention deficit hyperactivity disorder. Effects of parenting on self-regulation in adolescence appear to be moderated by Plasticity alleles, of which the 2R and 3R alleles of MAOA are 2, with the more plasticity alleles males but not females carried, the more and less self-regulation they manifested under, respectively, supportive and unsupportive parenting conditions. Depression. MAOA levels in the brain as measured using positron emission tomography are elevated by an average of 34% in patients with major depressive disorder. Genetic association studies examining the relationship between high-activity MAOA variants and depression have produced mixed results, with some studies linking the high-activity variants to major depression in females, depressed suicide in males, major depression and sleep disturbance in males and major depressive disorder in both males and females. Other studies failed to find a significant relationship between high-activity variants of the MAOA gene and major depressive disorder. In patients with major depressive disorder, those with MAOA-GT polymorphisms 6,323 rupees coding for the highest activity form of the enzyme have a significantly lower magnitude of placebo response than those with other genotypes. Antisocial behavior. In humans, an association between the 2R allele of the VNTR region of the gene and an increase in the likelihood of committing serious crime or violence has been found. A connection between the MAOA gene 3R version and several types of antisocial behavior has been found. Maltreated children with genes causing high levels of MAOA were less likely to develop antisocial behavior. Low MAOA activity alleles, which are overwhelmingly the 3R allele in combination with abuse experienced during childhood, resulted in an increased risk of aggressive behavior as an adult, and men with the low activity MAOA allele were more genetically vulnerable even to punitive discipline as a predictor of antisocial behavior. High testosterone, maternal tobacco smoking during pregnancy, poor material living standards, dropping out of school, and low IQ predicted violent behavior are associated with men with the low-activity alleles. The low-activity 3-repeat allele variant of the MAOA gene has also been found to occur frequently in men who join gangs. According to a large meta-analysis in 2014, the 3R allele had a small main effect on aggression and antisocial behavior, even in the absence of other interaction factors. Aggression in the warrior gene. A variant of the monoamine oxidase A gene has been popularly referred to as the warrior gene. Several different versions of the gene are found in different individuals, although a functional gene is present in most humans, with the exception of a few individuals with Brunner syndrome. In the variant, the allele associated with behavioral traits is shorter 30 bases and may produce less MAOA enzyme. This gene variation is in a regulatory promoter region about 1,000 bases from the start of the region that encodes the MAOA enzyme. When faced with social exclusion or ostracism, individuals with a low-activity MAOA gene showed higher levels of aggression than individuals with a high-activity MAOA gene. Low-activity MAOA could significantly predict aggressive behavior in a high-provocation situation, but was less associated with aggression in a low-provocation situation. Individuals with the low-activity variant of the MAOA gene were just as likely as participants with the high-activity variant to retaliate when the loss was small. However, they were more likely to retaliate and with greater force when the loss was large. Monoamine oxidases (MAOs) are enzymes that are involved in the breakdown of neurotransmitters such as serotonin and dopamine and are, therefore, capable of influencing feelings, mood, and behavior of individuals. According to this, if there was a mutation to the gene that is involved in the process of promoting or inhibiting MAO enzymes, it could affect a person's personality or behavior and could therefore make them more prone to aggression. A deficiency in the MAOA gene has shown higher levels of aggression in males, which could further stimulate more research into this controversial topic. A deficiency in monoamine oxidase A (MAOA) has been shown to be associated with aggressive behavior in men of a Dutch family. Legal implications. In a 2009 criminal trial in the United States, an argument based on a combination of warrior gene and history of child abuse was successfully used to avoid a conviction of first-degree murder and the death penalty. However, the convicted murderer was sentenced to 32 years in prison. The results showed the effects of the 4-repeat allele of MAOA promoter polymorphism on physical aggressive behavior for women. It seems that there is an interaction between the 3-repeat allele of MAOA promoter polymorphism and emotional abuse experiences on aggressive behavior for women. Epigenetics studies have linked methylation of the MAOA gene with nicotine and alcohol dependence in women. A second MAOA-VNTR promoter, P2, influences epigenetic methylation and interacts with having experienced child abuse to influence antisocial personality disorder symptoms, only in women. Animal studies A dysfunctional MAOA gene has been correlated with increased aggression levels in mice, and has been correlated with heightened levels of aggression in humans. In mice, a dysfunctional MAOA gene is created through insertional mutagenesis called TG8. TG8 is a transgenic mouse strain that lacks functional MAOA enzymatic activity. Mice that lacked a functional MAOA gene exhibited increased aggression towards intruder mice. Some types of aggression exhibited by these mice were territorial aggression, predatory aggression, and isolation-induced aggression. The MAOA deficient mice that exhibited increased isolation-induced aggression reveals that an MAOA deficiency may also contribute to a disruption in social interactions. There is research in both humans and mice to support that a nonsense point mutation in the 8th exon of the MAOA gene is responsible for impulsive aggressiveness due to a complete MAOA deficiency. Interactions transcription factors A number of transcription factors bind to the promoter region of MAOA and upregulate its expression. These include, SP1 transcription factor, GATA2, TBP. Inducers synthetic compounds that upregulate the expression of MAOA include valproic acid, Depakote inhibitors. Substances that inhibit the enzymatic activity of MAOA include, see also monoamine oxidase B. References Rehan W., Sandnava N.K., Johansson A., Westberg L., and Santila P. 2015. Effects of MAOA genotype and childhood experiences of physical and emotional abuse on aggressive behavior in adulthood. Nordic Psychology. doi 10.1080, 191276.2015.10269222. Further reading.